This is an A-level biology presentation looking at gas exchange in fish. In this presentation, we're going to look at the structure of fish gills, and we're going to talk about what's a counter-current flow system and how it means that the fish gills are adapted to maximize the rate of gas exchange. First thing I'd like you to do is to draw a diagram showing a fish and label the fish. I want you to spend no more than 1 minute on it. Do not spend more than a minute. Pause the presentation now, have a go, and when you're ready you can restart. Well folks, remember to read the question. So you had to draw and label a fish. Well, that circle has been drawn and labeled as a fish. Remember, don't make assumptions in questions, don't make them overly complicated, don't start jumping 15 steps down a path. Read the question and do specifically what it asks. You were asked to draw and label a fish. When we do this in science, we can draw any shape and label it accordingly. We were not asked to draw an artistic drawing of the fish or anything complicated than that. So that's why I said only do a minute on it. We didn't want to spend 20 minutes on learning all the external features of a fish. And you'll be lucky or unlucky to know that you don't need to know all of those things for the A-level biology course. However, should you wish to know all the labels and you have gone and done all the labels, just in case, we have added all the labels in for you and you can certainly check them off. But you do not need to know the external structures of a fish. You do, however, need to know that water goes in the mouth and the water comes out the little openings on the side, which are the gill coverings. And that is all you need to know. Remember, when we talk about gas exchange, and we've done a lesson on this at the start, if you haven't seen it, go back over the presentation on gas exchange, but it's all about Fick's law. It's all about increasing your surface area, increasing your concentration difference, and making sure that the distance, the exchange distance, is the lowest possible. And again, you need to remember that organisms need to get a balance between allowing gas exchange to occur, but not excessive water loss. However, now we're talking about fish, so excessive water loss doesn't play such a big role because they're obviously surrounded by water. We're now going to start looking at the structure of the gills. And on the goldfish, you can see the circle represents the gill bar, and the lines coming off represents the filaments, and you end up with this kind of pointed head, so the water would be going in the mouth of the fish and out of the gill plates. What we have inside the gill bar is two tubes, one carrying oxygenated blood and one carrying deoxygenated blood. Along the gill filaments, we also have little disc-like structures called gill lamella, and the gill lamella are at right angles to the gill filaments. If you look at the diagram on the top-down view, you can see that the deoxygenated blood arrives through the gill bar, it goes into the gill filament, and the gill lamella act like a capillary network that you would find in an animal organism. The blood then is oxygenated and it returns back along the gill filament and into the gill bar to be circulated around the body. When you start zooming in and looking at the gill lamella, this is what the kind of structure you'll see. You can see that on one side we have the deoxygenated blood being delivered, and just as I said about the capillary network and the gill lamella, we then have gas exchange occurring and oxygenated blood being fed back along the gill filament and back into the gill bar. So how are the gill lamella adapted? Well, they have a large surface area to maximize the rate of gas exchange, and they're also just a few cells thick. They're very thin, containing large numbers of blood capillaries, just like the capillary networks you would find in animals. Again, it all goes back to Fick's law and look at surface area, concentration difference, and the distance that the substance has to travel, and these are the things that will always be visible when you're looking at adaptations for gas exchange. So the question is, in what direction will the water flow and why? Pause the presentation, come up with your best answer. It's either direction 1 or direction 2. You've got the two diagrams and 1 and 2 match up on both diagrams. So your answer is number 1 and number 2, and then try and think of a reason why. Pause now and restart when you're ready. Well, in what direction will the water flow? Well, the water will actually flow in the direction of 2. It'll go from the oxygenated side across the gill lamella to the deoxygenated side. Now the question is why, and the reason is that it maximizes the concentration gradient for the longest period of time. And we're going to have a look at what that means. If you look at the bottom diagram, what we've done is we've taken the assumption that number 1 was correct. It's not correct, but let's assume it was. If number 1 was correct, what that would mean is when the deoxygenated blood arrived, it would be next to the fresh water that was coming in, and that water would have a very high concentration of oxygen in it. Hence, if you look at the diagram at the bottom, you can see that the water on the outside of the blood capillary has a concentration of 100, whereas inside the blood capillary there's very little oxygen and it's only at 10. As we move through the capillary, what happens is that the oxygen diffuses into the blood capillary. Hence, the concentration of oxygen inside the blood capillary is increasing, and the concentration of oxygen in the water outside of the blood capillary is decreasing. And that's fine until we get to the point where we hit equilibrium. And equilibrium, remember, is where the rate of diffusion is equal in both directions because there is no difference in the concentration. As a consequence, what happens is that we only end up with about 50% of the saturation of the blood. There's a lot more space for a lot more oxygen, but because of the concentration gradient, that oxygen will not move into the blood capillaries. So while initially we had a huge difference in our concentration gradient and we had a fast rate of diffusion, we actually have lost out overall because the oxygenated blood is nowhere near capacity. Now we refer to that as a parallel flow because the direction of water and the direction that the blood are traveling around the gill lamella are parallel to each other. And I'll say again, this is not really what happens. It is wrong, but we're talking you through to explain why this doesn't happen. And it'll maybe make a little more sense when you see what does happen. Well, what does happen is what is called a counter current or counter flow system. And it means that the direction that the blood is flowing is in the opposite direction to that of the water. So again, you can see the direction the fish is swimming from right to left. The blood is flowing in the same direction as the fish is swimming, whereas the water is flowing in the opposite direction. And if you look at the values on the diagram, you can see quite clearly that the difference in the concentrations is much lower. Instead of 100 to 10 initially, we actually have 100 to 90. So the rate of diffusion initially would actually be slower. However, all the way along the entire length of the lamella, the concentration of oxygen is always higher in the water and always lower in the capillary. And as a consequence, we always have movement of oxygen and net movement of oxygen by diffusion into the blood capillary. So by the time we get the end of the lamella, we have a much higher oxygen concentration in the blood compared to if we'd done the parallel flow. So the countercurrent system is the actual system that's used to maximize the amount of oxygen that is absorbed. It may not be as fast, but we get a lot more oxygen in there. And that's a comparison between the two, just to help you be clear in your head. You will be asked about a counterflow system if they talk about fish in the exams, and they will ask you why they use a counterflow system instead of a parallel system. And you've got to be able to talk about the fact that it maintains a concentration gradient throughout the length of the lamella in a counterflow system, whereas in a parallel system we get to equilibrium and we end up that we don't have the maximum rate of oxidation of the blood. So in this presentation we looked at the structure of fish gills and we looked about how they're adapted to maximize gas exchange. You need to know the structures, because if you don't know the structures you're not going to be able to talk about what's happening in terms of gas exchange very easily. So take the time to learn the structures first. Once you can name them, then you can start going on to explain how they're adapted.